So today we are going to study about the transgenic animals and targeted genome modification. As you know, transgenic animals are the animals which contain the foreign genes for any specific purpose, and how we can generate them, how we can modify their genome, that is what we are going to study today. So gene targeting, how we can target the gene by the homologous recombination method. So this is a very simple method where we are isolating the blastocyst. It means blastocysts are from the germ cells. You isolate the blastocyst and you take the inner cell mass of these blastocysts and you culture them. After culturing them, you introduce the foreign gene in these cells. And how you can do it? You can do it by the homologous recombination. For example, this is your ES cell that is containing this particular gene. This is the left side sequence, this is the right side sequence. So you have to make the vector that has this particular gene on the left side and this particular gene or the sequence on the right side, so they are complementary to each other. And now, because after the recombination, what is going to happen, this particular gene X is going to be integrated in this particular site. And where is this site located? It is present in these particular embryonic stem cells. Now you know that these are the, now you have to select, because not all of these cells are going to uh, show this HR homologous recombination with your vector. So there will be some specific cells which will show this homologous recombination. You have to select these cells. So how you can select it? This is like, either you can do the positive selection or you can do the negative selection. In case of the positive selection, your gene is tagged with the neomycin resistance marker. So whatever the ES cells, if you will grow them in the in the presence of neomycin, they will grow. For example, if you see here, suppose this is your ES cell in which there is no homologous recombination take place. So what will happen? This particular cell will be not showing any resistance for the neomycin. So what will happen, if you will grow this cell in the presence of neomycin, it will die. But suppose if you have the ES cell that has shown the homologous recombination and therefore it carry the neomycin resistance gene. So if you will grow this cell in the presence of neomycin, this cell will grow. So like this you have to do the positive selection. So your cell is going to be neomycin positive but the TK negative. In case of the random integration, it is going to be both positive. So you have to select the cells which are basically neomycin positive and the TK negative. Or it could be any other marker also, depending on the vector which you are using. So once you select the gene targeted ES cells, you will grow them. Once you grow them, you will inject into the another blastocyst and you will now inject these blastocysts in this particular mice. So after this is going to produce the chimeric offspring. Now you multiply this chimeric offspring with the female mice and now you will get the heterozygous mutant mice. So that's how you can target these particular genes. So here, if you see, what is this chimeric mice? Means some of this blastocyst cells, some of the blastocyst will be host specific, Okay, and some of the blastocysts are going to be derived from this mutant ES cells. So this is the definition of this chimeric offspring. So not all the cells are going to be to carry the mutant ES cells. Now coming to the chimeric, this is the chimeric offspring which you got, where not all the cells are derived from the genetically modified ES cells. Some are going to be derived from the host, and some are going to be derived from the genetically modified ES cells. Now you multiply this male chimera with this particular female mice, and you are going to get the heterozygous mutant. Means, what does it mean? It will carry one allele which is normal and one allele which is mutant. So here if you see, one allele is going to be normal and one allele is going to be mutant. But all the cells carry that mutant allele. So that is the difference. Here all in this particular mice, if you see, all the cells are going to carry the mutant allele. In this mice, not all the cells are going to carry the mutant allele. So this is the difference between this chimeric offspring and the heterozygous mutant, particularly in this figure. So what is the ultimate logic? The logic is how you can integrate this particular gene X in this mice so that it is going to produce after so many generation, it is going to produce the heterozygous mutant where every cell will contain the mutant allele. So that is the gene targeting. If this particular gene is of interest for you, so this mice is going to show all the functions related with this particular gene, and then you can study it. Now gene knockout, as you know, you have to inactivate the gene. Once you inactivate the gene, the objective could be to model a human disease caused by a loss of gene function, or simply to investigate the function of that gene. For example, if you are inactivating a gene in a particular animal, then you will not see the functional expression of that particular gene, and then you can study so many things, the mechanism and other things, to see what could be the relevance of that gene to that particular disease, or for a particular mechanism. So this is the gene knockout. In case of the gene knock-in, you are expressing that particular gene. For example, if you see here, EBC is a gene which is having 21 exons, 1 to 21 exons, and uh, the inactivation of this particular gene leads to the Ellis-Van syndrome. What happened in this particular syndrome? The child has a very small fingers and defective craniofacial features. So this gene is, so scientists want to study the relevance of this gene. If you inactivate this gene, what will happen? Like that, they want to study. So this particular gene has the 21 exons. So what they did, and the exon 1 is critical for the expression of this particular gene because it has the translational initiation codon methionine, which is ATG here. So what they did, basically they integrate the LAGG gene in place of this uh, uh, these many exons. They integrate the LAGC gene along with the neomycin marker. So now this LAGC gene is with the promoter of the EBC gene and with the ATG codon. Rest of the exons, rest of the uh, sequences were deleted because of the integration of the LAGC gene. So let's say this is exon 1, this is exon 2. So this is the initiation codon and this is the promoter. So you will see they integrate the legacy gene, so this particular thing is going to be deleted, and they tagged it with the neomycin marker. So then they study the expression of this particular gene in this particular, I think it is the mouse. In mouse they did this experiment. Okay. So they can see where this particular gene is going to be expressed. So they found it in the maxilla. As I said, this is critical for the skeletal development. This is critical for the limbs development, even in the human being. So they want to study the relevance of this gene, and that is why they integrate the lexi gene along with the neomycin marker. So you can see the expression images show the beta galactosidase activity in the EBC plus minus embryo and represent expression in the whole embryo. So where this particular gene is expressing, and if it is expressing here, what is its role? So they want to study it. Okay. Okay. Now the very important example is the Cre-Lox system. This is the system originate from the bacteriophage P1, which is a virus that infect the bacteria. So there is a Cre recombinase enzyme which recognizes in this Cre-LoxP system. One is the Cre recombinase enzyme that recognizes a particular target sequence. And what is this target sequence? If you see here, this is I think this is 32, 13 base pair repeats. These are the 13 base pair repeats you will see. ATAA, so you can see ATAA, C double T like this, and similarly here TATT, so you can see TATT. So these are the repeats, and these are the 8 base pair sequence. So whatever the sequence present in between these repeats plays an important role in the either the gene is going to be deleted or gene is going to be inverted. So this particular sequence plays an important role if gene is going to be deleted or it will be inverted. So for example, if you see here, if suppose this is your repeat sequence, 13 base pair repeats, this is your sequence GCA, TACA, T, and then again the 13 base pair repeats, and then there is your gene which you are targeting, and then again there is going to be the 13 base pair repeats. And suppose the same sequence if you are getting GCATA, CAT, and then again the 13 base pair repeats, because they are in the same orientation, these 8 base pair sequences, because they are in the same orientation, gene is going to be deleted. Same orientation means gene is going to be deleted. What happened in case of if they are facing each other? For example, this is your 13 base pair repeat. If this is your 13 base pair repeat and you are getting this sequence GCATACAT, then 13 base pair repeat, then there is your gene, and then again your 13 base pair repeat, and then the sequence is, let's say if it is TACATACG and 13 base pair, then gene is going to be inverted. So you can see here the 8 base pair sequences are playing very important role. Here if you revert it, ATACG. That's what I wrote here. So if they are facing each other, if they are facing each other or they are in the reverse orientation, then the gene is going to be inverted. So this particular sequence, if we talk about this particular 8 base pair sequence, plays a very important role whether the gene is going to be inverted or deleted. So this is the Cre-Lox system. Here you can see, here the scientists have generated a mice that has the LoxP sites as well as the gene. Okay, so there are two LoxP sites and there is one gene. So this mice is basically LoxP mice. They also created a Cre mice that has the Cre recombinase enzyme in the presence of specific tissue specific promoter. Now what will happen, this Cre recombinase enzyme will recognize these COXP sites, and based on the orientation of these LOXP sites, if they are in the same orientation, the gene is going to be deleted. For example, in this case it is going to be deleted, but if suppose it is going to be in the reverse orientation, and when we are talking about reverse orientation means only the 8 base pair sequences. If these sequences are in the reverse orientation, as I showed here, this particular 8 base pair sequence, if it is in the reverse orientation, then the gene is going to be inverted. So this is the Cre-Lox system. Now we are coming for the zinc finger nucleases, by which you can also inactivate the gene. uh, this is the zinc fingers. There are 4 zinc fingers. They are tagged with the DNA cleaving domain monomer. Each zinc finger recognizes the 3 base pair sequence, so it will recognize the 12 base pair sequences. So this is the left ZFN recognition site. Then there is going to be the right ZFN recognition site that will recognizes the right side bases. This will recognizes the left side bases. Okay, you have to create these domains like that so that only the heterodimers are forming, not the homodimers. Homodimers means the same 1 2 3 4 is not forming, is not going to bind to the site here again, and it is going to form the homodimer. We don't want the homodimers, we want the heterodimers to form. Once this heterodimers form, this particular sequence is going to be cleaved, and after the cleavage, as you know, there is either the non-homologous end joining repair or the homologous repair is going to be there. So here you can see here, so why these nucleases are called zinc finger, because they have the zinc molecule in them, and uh, they have the DNA binding domain as well as the cleavage domain. So DNA binding domain will bind to the DNA and the cleavage domain will cleave the DNA. Here the important thing is still this: the cleavage domains of the two ZFN can be designed to be asymmetrical, so the heterodimers form and not the homodimers. So only the heterodimers will form, then only the cleavage is going to be. You have to design your experiment, or you have to design these cleavage domains like that so they are not joining with each other. So this is the zinc finger. Here you can see the ZFN each have 4 zinc fingers and designed to recognize the 12 base pair sequence, because one zinc finger will recognize 3, so 4 zinc finger will recognize 12 sequence, 12 base pairs. So they will recognize the 24 nucleotides, 12 on the left side, 12 on the right side. Then the DNA cleavage domain will cut it, okay, once they dimerize. So this is the zinc finger nucleases. That's all for today's lecture. Just go and study about this EVC gene and try to see pictures about this particular syndrome where the children are having very small limbs and special craniofacial features. Their skeletal is also not developed, and that was the reason why the scientists want to study this gene, and that is why they put the LEGC gene, so they can see where the expression of this gene is more and where the expression of this gene is less. So that is why they integrate the legacy gene along with the neomycin marker. So that's all for today's lecture. Thank you.